All right, in this lecture, we are going to continue where we left off, and we are going to continue to look at our sugars, which is our third major molecule we're looking at this semester. Just as a reminder what those were, we started with DNA, which is our instruction manual to make everything. Then we looked at proteins, which are the major players in the cell, being our catalysts and our structures. Now we are moving on to sugars. And our last slide on the previous lecture was we are looking at disaccharides and how we name these linkages when we combine two sugars. Now we're going to specifically talk about the different types of polysaccharides that we have in our body. And just as a reminder for the word I just used there, polysaccharide. Excuse the spelling, that's not even close. But poly means many, saccharide means sugar. So it's just many sugar compound. And so we're going to look at the most abundant sugar there is, and that are saccharide, and that is cellulose. And this is what's making up your plant cell walls. And cellulose is just two glucoses linked together, and it's beta 1,4. Let's take a look at the linkage. Right, so we find our anomeric carbon, which is right here, and that's carbon 1. This is going up. The CH2OH is going up, so that's beta. So that's why it's beta 1. And then here's our anomeric carbon, 1, 2, 3, 4. So this is beta 1,4. And beta 1,4, we cannot break down. We do not have the enzymes for that. Termites, cows, they do have bacteria in their guts that have the enzymes to break that down. Humans don't. Be a lot easier if we could, but we can't. And so for the cellulose, as I have here on the slide, you got roughly 15,000 glucoses in one line. But what makes cellulose really strong is hydrogen bonds. So what happens is that, you have a sheet that's 15,000 glucoses. And next to that, you have another sheet. Below that, you have sheets as well. And these blue lines are showing you all the hydrogen bonds. So the hydrogen bonds are being connected to the sheets below, sheets above, and the sheets next to you to form this giant hydrogen bond web, which makes cellulose so strong, right? That's why it's basically one solid meshwork, and all it is sugar. It's kind of hard to believe that when you think about it, that it's the same stuff we eat normally, like table sugar. The only problem is we can't digest it because of this glycosidic bond, right? And like I was telling about, you need an enzyme to do that. The enzyme's called cellulase. There's also another type of sugar that is used for structure called chitin. That's also a beta 1,4 linkage. And it also uses glucose, but it's a glucosamine. So at position 2, we have this acetyl group, this N-acetyl group on our chitin. But otherwise, chitin and glucose look very similar. Just the difference at the second prime position. And chitin, that's what you find on beetle shells. All right, that hard exterior is chitin. It's just glucose again, just a sugar. So this problem right here is to help test that you know what the structure of cellulose looks like. So try not to look at the glucose picture on the back. Try to take a couple minutes, try to figure this one out yourself. If you can't get it from your memory, if your brain's still a little foggy starting this video off, then go back. But yeah, let's take a look at the answers to this. So cellulose treated with methanol. You methylate free anomeric carbons. How many methyl groups would be incorporated per cellulose chain? So I'm going to go back to the picture for this. So our anomeric carbons are right here. and we have roughly 15,000. And I'm just going to draw the last glucose. So at the end of every chain, you're just going to have a glucose that looks like this, because the chain has to end eventually. So this is a free anomeric carbon. This is our reducing sugar. So out of every chain of cellulose, you have exactly one reducing sugar, one free anomeric carbon. It's the one at the end. So going to our question, how many methyl groups will be incorporated per cellulose chain? That is 1. And then B, if you treat it with dimethyl sulfate, how many methyl groups do you add to free hydroxyl groups other than the first and last glucose? So let's take a look at this. How many free hydroxyl groups do we have for our glucoses and cellulose? We have 1, we have 2, we have 3. So it should be 3. So that's the overall structure of cellulose. I'm looking at the monomer level. Okay, let's go to a sugar we can eat, and that is starch. So starch is the main energy reserve of plants. So when they're doing photosynthesis, they'll store some of that energy as starch, and we can eat starch. And I find it amazing myself that the only difference between starch and cellulose is that in starch, the linkage is alpha 1,4. And cellulose is beta 1,4. And that alpha to beta switch, which again, the only difference is which way the OH on the anomeric carbon is pointing. On the alpha version, you get ribbons. On the beta version, you get flat ribbons. So that's just one difference that allows us to eat or not. And starch is made out of 2 different chains. One's called amylose, one's called amylopectin. And let's take a look at the differences between the 2. So amylose has no branch points. What I mean by that is that it's this blue that I'm looking at here. That is alpha 1,4 from start to finish. That's it. Amylopectin does have branch points. What I mean for that is that your glucoses are going to be connected to alpha 1,4 until eventually one of your glucoses rather, and that's being actually shown down here, you're going to make a 1 to 6 branch point where you're going to branch off into a new sugar or a new chain as being shown here. So here we had alpha 1,4 and it's going until it reaches this point where it's being branched. So at the branch point where we have this alpha 1,6, we had one sugar making a 6, and it's also making alpha 1,4 to its previous neighbor, and it's making alpha 1,4 to its next neighbor. And what this does, what this allows us to do is to access the energy from starch incredibly fast while keeping it somewhat compact. Because for starch, the way we release glucose molecules is through the non-reducing end, right? So plants have an enzyme that will go and cut one glucose off a non-reducing end. So by doing this branch structure, you have a lot more non-reducing ends. Compare that to reducing ends, you only have one reducing end. So in our example here, we have I forgot amylose up here. So we have 2 sugars that are called reducing ends. So if our enzyme cut sugars off reducing ends, we get 2 glucoses per event, which is slow. Because if we cut from the non-reducing ends, we get 1, 2, 3, 4, 5, 6, 7, 8. We get 8 glucoses, right? So we get a 4-fold increase just in this picture. And in reality, it's way bigger than 4. But just in this picture, we have 4 times as many glucoses being accessible if we cut them off the non-reducing end than the reducing end. So this branching is allowing us to create a lot of non-reducing ends for our enzymes to cut off glucoses that plants can use for energy. For animals, for us, we have something like starch, but it's called glycogen. And this sugar storage is going to be in your skeletal muscles and your liver. The majority of the glycogen is actually in your liver, because one of the main job of your liver is controlling the amount of glucose that's going in and out of your blood. It's a big glucose storage molecule. And it's very similar to amylopectin, except glycogen has a lot more branches. Glycogen is branched a lot more. Again, this is for storage and also to access the energy fast. The enzyme that breaks down glycogen is called glycogen phosphorylase. And those of you taking Biochem 2, we're going to see this enzyme again. And depending on our speed, we might again revisit glycogen phosphorylase in Biochem 1, because it is a good model enzyme for seeing how enzyme control happens. But what glycogen phosphorylase does is that it's just like what I was talking about, where if we have a lot of branches, right? So branch, 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 you know, something like that. We're always going to cut from the non-reducing end, because every time we make a branch point, we're making a new non-reducing end, while we'll only have one reducing end. So if your body is in a state of starvation, you can get glucoses out a lot faster into the bloodstream if you cut from the non-reducing end. Also, a way to speed this, or a way to access the glucose, is through glucose debranching enzyme. Because when you are breaking down glycogen, like you are releasing glycogen or glucose in the body, glycogen phosphorylase can't work roughly 6 glucoses from a branch point. So when you're 6 glucoses out from a branch right here, glycogen phosphorylase stops working. It can't cut off any more glucose. So what debranching enzyme does, it will come and it will break this branch point. It'll break this 1,6 bond. When it does that, this will release our 6 long glycogen molecule, and now debranching enzyme can work, and it'll cut off the rest of the glucoses that we can access. So that is how your body has short-term glucose storage. Keep in mind though, you have about 1 day's worth of glucose energy in your body. You don't store much glucose. Your energy storage is from fat. All right, again, that's something we dive into biochemistry 2 more. All right, so here's just some questions to test your knowledge again. Take a minute, read them over, pause the video, and I'm going to jump into the answers right now. All right, so we have a molecule of glycogen, 10,000 residues. It branches every 10 residues. How many reducing ends do we have? So you might be tempted to do some math of like 10,000 divided by 10 and get the answer of 1,000. The answer is 1. Right? Remember, the reducing end is the very last glucose. You're always only going to have 1 reducing end in glycogen, an amylose, and an amylose pectin. Here's B. It's a little more tricky. Is amylose or amylopectin likely to be long-term storage? And I'm going to go back to the images for that. So amylose is blue. It doesn't branch. Amylopectin does branch. And so the correct answer here is amylose. The reason for that is that you can make a very, very long amylose, right? Amylopectin branches. So its length is somewhat limited, and you're only going to be cutting one glucose off at a time. When you're only cutting one glucose off at a time, it's going to take a long time for this amylose to be eaten up. For amylopectin, it's many branches, and every branch you're taking one off. So this is going to be eaten up way faster, your amylopectin. So the amylose is slow release, right? Slow release glucose. And that is why it's storage. It's just because, let me see if I can explain this a little better, why it's storage. When you turn on the enzyme to chop off glucoses from the end of amylose or amylopectin, there's no way to tell that enzyme, don't touch one of these molecules because I want it as my safety reserve. The enzyme's not that smart. Your cell's not that smart. But what happened is that we created, or plants created rather, a different type of molecule to ensure that slow release mechanism, where the plants just said, I'm going to create one glucose polysaccharide that doesn't branch. When I do that, now my glucoses aren't going to be all cut off very quickly. It's just going to be one at a time. And if I have 10,000 glucoses in that amylose, and I cut one glucose for every 1 second, which is slow, but it gets the point across, it's going to take me 10,000 seconds to release all my glucose, which is, we're talking about hours at that point. So it's a good way for plants to have a slow, steady stream of glucose, your long-term storage. All right, talking about some more disaccharides, we have our glucosaminoglycans. Here, I am not looking for you, and I'm going to repeat that in case you're listening to me at 1.5 speed or 2 speed, probably more likely, not needing to memorize structures here. So I have 6 glucosaminoglycans that are examples, but I'm not going to ask you on the test, like what does chondroitin 4 sulfate look like, and give you a picture. But what these molecules are, in the name aminoglycan, you have taken one of your hydroxyls and you put an amine group there. Also, for glucosaminoglycans, not only is 1, they have amines, 2, they are negatively charged. At another OH location, you have put negative charge. So these are, they have amines, they're very negatively charged, and 3, they are repeating disaccharide units. Those are the 3 characteristics, like the basic characteristics of glucosaminoglycans. And what these do is that they are in your extracellular space. So these sugars on the outside of your cells, and you are going to find them in your connective tissues. They're going to be interacting with collagen and different proteins in a big old matrix. And these sugars, when they're on the outside of your cell, they form a very slimy and mucus-like substance, very viscous and elastic, which are good for like your joints, right? So in your joints, you're going to have a lot of these glucosaminoglycans because they form this mucus gel-like substance, which is great for shock absorbers. And also, this last bullet point here, some of these are sulfonated to make more negative charges. And also, next time you're in vitamin aisle, if you see a bottle called chondroitin or glucosamine, it's basically just these sugars. The idea there is that you take these to try and improve joint health, right? Although, as far as I know, there's no clinical study that actually shows definitively that they do that. It's just, we know that they're there. So people think, well, if I eat it, maybe it'll replace itself there. But so far, the jury is out on the benefits of that. And we have one more problem on this slide before I jump to the next. Again, just checking your mastery, and go back to these problems before the exam to make sure you understand everything here. What is the net charge of chondroitin 4-sulfate for a molecule containing 100 disaccharide units? So I'm going to answer here. So if you haven't figured it out yet, please pause. So chondroitin 4-sulfate has 2 negative charges. And if I have 100 disaccharide units, so 1 disaccharide is -2. So 100 would be -200. Now let's talk about its usefulness inside your body. And fibroblast growth factor will bind to these glucosaminoglycans in the EC extracellular matrix. What amino acids do you think will be there? And you thought we were done with amino acids. You thought wrong. Amino acids are everywhere. And so these are negatively charged. So I would expect something that's positively charged to interact with these sugars. All right, let me switch PowerPoints here really quick. I don't know why I did that weird accent there, but here we are. Now, whoops, that was not the button. So let's talk about what are called glycoproteins now. Let's just break down that word. Anytime you see glyco, you're talking about a sugar. Anytime you see the word protein, you're talking about a protein. So a glycoprotein is a protein that has sugars on it. And the amount of sugars vary, right? It could be very light, where 1% of your protein's weight is due to sugars, or it could be mostly sugar and a few protein. 90% of the weight's due to sugars. And what's really weird about glycoprotein, something that's really odd, is that it has microheterogeneity, which means that the carbohydrates that are placed on your proteins are variable. The reason why I say this is odd, because let's say you make 5 of the same protein. The amino acid composition of all these proteins are going to be the same. The different modifications are all going to be the same, but the sugars are going to be different. So let's say you have a triangle there. Here you have a triangle, circle, circle, circle. Let's say you have like a square triangle, circle, triangle, where like triangle, circle, square are all different sugars. So this is microheterogeneity. You have the same copy of the protein, they can just have different sugars on it. And so the field of glycomics, which is the study of carbohydrates, is actually, it's quite difficult. Those of you who are interested, Dr. Ali here, Texas A&M San Antonio, he is doing research on glycomics. More specifically, his research is looking, I believe, at Parkinson's disease and these glycoproteins. So if that sounds something interesting to you, reach out to him, see if you can join his research efforts. But let's go back to the topic at hand. So we have these things called proteoglycans. And what a proteoglycan is a protein plus these glucosaminoglycans we talked about in the previous slide. And these are going to be hanging out in your extracellular matrix. And these are, again, they're going to be very viscous, very gel-like, again, for your joints. And these proteoglycans, they have a set structure. So let's take a look at this. Now, these are mostly sugars, right? So we have the main part of this is hyaluronate. And let me just, sorry, I just have to go one slide further. Okay, so I'm actually going to talk about these in the next slide because I do have a slide that goes more into depth on them. So let me do that. So going back to our structure, right? So the main rod here is hyaluronate, which is something we looked at on our aminoglyco disulfides. So this is a sugar. You got this big old sugar that's roughly 50,000 repeating disaccharides or 100,000 sugar molecules. And at branch points, and this blown up image is really good. At branch points, you have what's called a link protein. So you have low proteins connected to this sugar. And connected to the sugar in the link proteins is what's called a core protein. So this core protein is just another protein. And off of this core protein, you have more sugars. So at the very beginning of the core protein, you have keratan sulfates. And then later on, you have chondroitin sulfates. And so what happens, if you look on the picture down here, which is actually a real image of our proteoglycan, you get this big, bushy branch structure. It's almost like a pipe cleaner. If you don't know what a pipe cleaner is, it's like a little metal rod that's full of little bristles that you can use to clean small pipes. So it's very much like that. And how do we connect our hyaluronate to our proteins? We do that through our friend, the glycosidic bond. We talked about that on Wednesday's lecture. And for proteins, there's only two, well, for glycosidic bonds, there's only two ways to link. That's either N-linked or O-linked. For proteins, if you're N-linked, that means you have to be connected to an asparagine. If you're O-linked, that means you can be connected to a serine or threonine. So that is how we're making our sugar protein connection in our proteoglycan. And that's how you make any sugar protein connection, really. It's either going to be connected to an asparagine or serine threonine. Now, these hyaluronates, really big, 4,000 to 40,000 angstroms. An angstrom is 0.1 nanometers. So this is 400 to 4,000 nanometers. And I'm sure for you that you don't have a scale of what that actually means. But this is way bigger than the majority of the proteins in your cells. Like your proteins, you know, you're looking at 100 nanometers, right? 1,000 angstroms. Right. So, you know, this is 4 times bigger than like your average protein. And off these, so we talked about the core proteins, that's these branches. You have about 100 of those. Each of those core proteins, you know, 50 of the blue sugars, 100 of the red sugars. So this picture is a little poopy. It should be, you know, 1/3 blue, 2/3 red. And like I said, these are very hydrated. So here we have our collagen that's interacting with our core proteins and our sugars, all connected to our hyaluron. And you're just going to form this big old gel, for example, on your knees. That's why you can move around and jump and your knees don't explode, because this is a shock absorber, right? This gel-like matrix. So here's our first question for us. To do some critical thinking on how all this works inside your body, inside your cells. Read these and then come back and listen to my answer, double check. And I'm going to jump into the answer right now. So following their synthesis, many proteoglycans are highly condensed. These compartments also have many calcium ions. What are these calcium ions doing? All right, to really understand this, you need to remember what the structure of our chondroitin and keratin are. Remember, I already talked about this in this lecture. What you need to take away from our sulfates are negatively charged. And so if I want to take this giant molecule and wrap it into a ball, you're going to have all these negatively charged sugars repelling each other. So these calciums are acting as counter ions, right? They're going to be interacting with the negative sugars and helping them to be next to each other and not feel each other, right? So it's acting as a charge to help condense these. And then when we want to deploy this outside the cell, we don't want to be wrapped up in the ball. We want to be fully spread out. And so what's happening is that the CO2 gets pumped out, so it's no longer interacting with the sulfonate, and the sulfonate can spread out like, like an inflatable pool or an inflatable bed, where once you put air into it, it's just expanding. So here, once we remove calcium, it will expand. B, looks like a repeat here of a question just already asked. What type of amino acids do we want to bind to our glucosaminoglycans? That's positive, R and K. All right, question 2. This is a very good question. I like it a lot because, again, it's using information from what we talked about in the previous semester and synthesizing it with new information. So when you do SDS page, your glycoproteins usually come out as a big smear, while non-glycoproteins are a sharp band. So I have a bunch of smears here. So let's say all these are glycoproteins, while your normal protein will be a nice little band. Explain why that is. And if we think about this, so I'm going to jump into the answer now. Remember, glycoproteins have what's called microheterogeneity, which means that you can have multiple copies of protein, different sugars. And if they have different sugars, they have slightly different weights. Remember, SDS page separates on weights. So, for example, if we look at this band, maybe you have some copies of your sugar that are exactly 45, some that are 45.1, some that are 44.9, some that are 45.15, on and on and on, because they all have slightly different sugars, which means you're going to get a lot of markings all in the same area, leading to that smear-like appearance. While non-glycosylated proteins, proteins without sugars, the amino acid, the primary structure never changes. So they're all going to be the same weight regardless, right? And so you get this nice crisp band because every single copy would be 45. There's going to be no variability in that, so they're all going to appear at the same location. All right, I think, yeah, I'm gonna pause it here. We're gonna talk about peptidoglycan layer on Monday, talk about our gram-positive and gram-negative bacteria. But other than that, hope you enjoyed this sweet lecture on sugars. And I will see people on Monday. I'll put a homework up. If you have questions until then, let me know, and I'll try to get back to you when I can. All right, take care, everybody. Have a good weekend.